Again, what you see are just again oval shape single cells for the slide appearance. We have for Rhizopus, we've got the asexual and the sexual. So Rhizopus has a sporangium. So our stalk right there, our upright vertical hyphae called sporangiophore. Here is the Rhizopus sexual form, and what we look for are the zygosporangia structures here, and then you see the two different conjugating hyphae. So again, this is still Rhizopus, but this is sexual. All right, for Penicillium, we've got kind of vertically oriented conidia. So again, that part there, and then the structure supporting it, conidia 4. We've got Aspergillus plate. I won't show you the Penicillium. So again, I talked to you already about the two plates. Remember, Aspergillus has got the black-looking spores on mine here, and then the Rhizopus is real kind of light gray colored for the spore-producing structures. You can see that it's black on this one right here. Aspergillus itself has, again, three-dimensionally arranged conidia spores sitting into the environment. So again, you see the spores on the edge. Ours usually are pretty stained, real dark in the center, but you can see the little spores sticking off on the edge. And then let me remind you too, I have the dermatophyte picture, which is right here. Again, epidermal phyton for the example, the ringworm example, so make sure you see that one as well. Now the thing about the protozoans, we have here first of all the trypanosoma. Two different samples have blood, so you can see the red cells right here, but these of course are between the cells, they're flagellated. The vector's a tsetse fly. Again, that picture out here somewhere. See, it's full of blood. Here is the Trichomonas vaginalis. So kind of similar to the Giardia, except we only have one nucleus, but otherwise you see quite an array of flagella there. Here is the Giardia. So remember the two nuclei look like two eyes looking up at you, and again you've got flagella there on it as well, typically heart-shaped. All right, the Leishmania, which we didn't talk about in the lecture, but another example of a flagellate. And so again, you can see that pretty clearly, sandfly vector here for this one. The amoeba group is Amoeba histolytica. The Trophozoides means it's the one that kind of moves around, feeds, and so on. And then we have our cyst right here. The cyst has the 4 nuclei. This pattern where you've got the spot of DNA and the ring around it called a karyosome. And so anyway, that is characteristic of this group, and you can see we have the same thing back here on this one as well. So very distinctive for the amoeba group. All right, now for malaria, what we see are the merozoites now inside of the red cells. And again, red cells don't have a nucleus, so anytime you see a staining structure inside of there, you have something wrong. So anyway, again, you see on the inside now of the cells for the Plasmodium, female Anopheles mosquito, and remember for the 100th time, female and Anopheles and mosquito for our ID here on the vector. Toxoplasma, we've got the sporozoites here, and then showing kind of merozoite over there on the other side. I just used this picture right here if I'm going to show this one on the test. The Cryptosporidium, which again I got the picture over there. So the thing stains acid fast, kind of pink color, right? You know, and again part of the Apicomplexa group. And just close up. So on my picture there, one side had the previous one, on the other side is this one here, just again to show a little closer view of it. Bacillium coli, characterized by, again, a macronucleus, which is this very elongate structure here inside. And the cyst versus trophozoite, I don't usually ask you about that, but you can usually tell the trophozoite. You can see the cilia, the little array of structures around the edge, because cilia, whereas the cysts don't have any cilia. Just an example. All right, so as we go to the flatworm group, Norris here, Asian liver fluke. So what we have is the mouth end up here. There's an oral sucker. You can't really see it on this one, but there's a ventral sucker right there. We've got two parallel digestive tubes running along the length here of the body. This is the uterus part right here. This is the testy part. I don't remember now if the one we actually looked at in lab showed that or not. Egg showing here on the edge. But this is Clonorchis, again the liver fluke. The egg for Clonorchis again has this rather oval shape, but on this one end the opening, one operculum structure there. Okay, for the tapeworm, of course, we've got scolex up on the invisible end up right here. And then, of course, proglotid segments. And the ones at the very end would have the, of course, matured eggs. So tapeworm scolex. And so what you want to think about is that you can see these two sucker deals, almost like eyeballs looking at you. And then you've got the hooks here around the end. But that specific structure there for it is the scolex part of the tapeworm. And then for the proglotid, again, I use this picture right here. Mainly what we're seeing here, all of this is uterus type structure, and all of the testy parts kind of infused in the background. So anyway, tapeworm proglotid segment. Here's the tapeworm egg, so it's pretty circular. Got a big kind of rind looking deal around the periphery right there to protect it from the acidity of the stomach in the cattle. So anyway, here is our dog tapeworm. Now, obviously a lot littler, but you do have, again, a scolex region with two suckers and hooks. You've got a kind of neck zone, and then you can see we got, in this example here, 3 proglotid segments, right? So it'll be very, very small, short relative to the other one. The Echinococcus hydatid cyst structure. Got some pictures, a little better pictures of it over there, but what you have is this big circular deal. These black things on the interior are the inverted scolices, and the picture I have here is the one that you want to really make sure you get for that. It's clear right here, so one on each side. So make sure you take a picture of that. I would use that. I can't ask you to look at that on a slide, but it would be on something looking like that over there. With the schistosome, the male and the female, of course, connected together. Male is the bigger one, the female is the narrower one. The sort of cercari for the schistosoma looks like a little torpedo, basically, and so you've got a head region, and you can see the tail with the 2 little extensions right there. That's the one that would actually infect a human. The egg's absolutely the most easy to recognize because you've got the spine type structure that projects from the side of the egg. All right, and then here we get to what we're doing today. So Anaerobius vermicularis for the pinworm, they're looking specifically for this enlargement right here. I do have this picture right here, but I did put again back there an example of one with a big round esophageal bulb. It's the only one that has that characteristic. Not a very good picture of the egg, but the thing is you've got a flattened side and then a more pronounced curved side. This one happens to show the embryo effect, but we don't really see that on our slide. So you're looking for the kind of asymmetrical shape for it. Here's a scarus. Of course, over there I've got the 2 samples. So remember, the big one is the female, and the smaller one with kind of a bent end is the male. And then here is the egg for the scarus. You've got that kind of bumpy sort of appearance on the periphery right there. And then the hookworm. Again, I got that pink picture is actually better for this than this one, but you can see the mouth plate, so that's where it attaches. The Trichuris whipworm got over there on the scope, so again, the head end is actually up on this little tiny, and you can hardly see. And anyway, so again, fairly distinct in terms of its appearance relative to the others. Here is the egg, and again, 2 nice openings, 2 opercula on each end. And then here is the trichinella. So you see these cysts, and again, this red tissue in the background is muscle. It's typically target tissue, right? So that's the red.